So this background of a Costa Rican beach that we'll sort of start off with. And when I was thinking about what I wanted to present today, I was really trying to think back over not just the past 5 years, but my entire research life, and in fact, my entire life. And the first thought that I had is that maybe I should present something on insects. So I've spent quite a lot of my life studying insects in various forms. I've looked at how insects can be control agents for invasive weeds, how invasive insects can become pests of grapes and other small berries, how insect food webs assemble in managed grasslands, and the implications of coffee farming and tropical deforestation for insect biodiversity. But when I was thinking back on what I really wanted to talk about today, I started to think about all of the other different types of biodiversity that I also care about. I'm rather fond of plants and amphibians and giraffes and reptiles. And even recently, Gretchen and Nick have managed to convert me into a fan of birds. And when I was thinking about what I should talk about today, I really was struck by this recent report from the Intergovernmental Panel on Biodiversity and Ecosystem Services, which really crystallizes a lot of what we already suspected and knew was happening to our biodiversity globally. That climate change and habitat degradation and these other human-induced global pressures are driving species extinctions, not only for insects, which I'm so deeply fond of, but also for all of these other taxa as well. And so today I wanted to talk a little bit more broadly, and instead of focusing on just one taxonomic group of insects, focus on biodiversity patterns and dynamics across all of these different taxonomic groups, with a real emphasis on how climate and land use are shaping both current day patterns that we observe, but also how climate change and land use intensification are likely to affect things into the future as we continue to reshape our planet's surface. But before we look to the future, I actually want to start by looking to the past. Somebody who shaped a lot of my work over the past 5 years, Alexander von Humboldt, who's by many considered the father of modern biogeography. And he wrote this very influential book at the beginning of the 19th century, where he detailed his journeys through modern day Colombia and modern day South America, where he would often go up these volcanoes of here, Chimborazo, and these other places throughout Colombia, tracking the types of vegetation that he observed as he would go along with his team. And keep in mind that he is a German writing at the beginning of the 19th century, so I think the quote, "It would be interesting to show on botanical maps the areas of where these similar groupings of plant species live," was about as enthusiastic as he was likely to be, or that matter, any of his contemporaries would likely be. And he went on to say that as the appearance of the vegetation changes, so too does the form of the animals, the mammals living in the woods, the birds flying in the air, even the insects gnawing in the soil. And this has been a question that's really gripped modern ecology since Alexander von Humboldt. And I think it's most succinctly summarized by Robert MacArthur in his 1972 book, where he simply asks, do different plant species change synchronously, or does each have an independent distribution? If they change synchronously, vegetation types are more than a mere convenience. They are real and hence a necessary subject of study. There's obviously a lot of other thinkers that have been wrapped up in this quote, with Clements and Gleason certainly coming to the forefront in terms of their contrast in ideas of plant species being distributed as communities or according to individualistic properties. And then also Whitaker and Holdridge and all of these other macroecological thinkers. And this question that Robert MacArthur posed back then has still not succinctly been answered. And I'll get into the reasons for that later. But to phrase it simply and ask why should we care, my first research question that I'll address today is, do species in fact organize into geographically identifiable communities? And while we certainly will get more into the scientific and technical parts of this question, I want to actually start by thinking about my childhood once again and Miss Frizzle on the Magic School Bus. And when I was a kid, through these books and television shows, Miss Frizzle took us to the deserts and to tropical rainforests,
Colin, he is Miss
to the
Frizzle.
Arctic, and all of these different places, understand what types of plants and animals you were likely to find there. And the one thing that kept coming back to me throughout this whole thing is that there are animals that we know are rainforest affiliated animals. There are animals that are desert associated. So we have these distinct communities that are associated with these different ecoregions or these different biomes, these different places where we expect to find one community of animals associated with one type of habitat and one ecosystem. And from a scientific perspective, these ecoregions or other groupings of species in space have informed a lot of modeling. So here I'm showing a paper from 2018, which looked at the relationship between carbon and terrestrial biodiversity at an ecoregion level. So here, dividing the world into these distinct geographic communities, you see either positive or negative relationships depending on where in the world you are. And so, for example, here we see that there are ecoregions with strongly negative and strongly positive relationships right next to one another, which indicates that if there is some sort of real biological mechanism underpinning these relationships, it could be driven by the species. However, if there's not, then it's just sort of an arbitrary way in which we've divided up the planet to make our models fit better so we can get better p-values as scientists. And then also from a conservation perspective, these ideas of ecoregions or ecosystem types has been widely used. For example, the IUCN Red List of Ecosystems. Here I'm showing for the country of Costa Rica which ecosystems have been deemed to be under various levels of threat, with these red ecoregions being the most threatened and these green ecoregions being relatively unthreatened. And once again, you see this striking relationship where there are very threatened and very safe, relatively speaking, ecoregions right next to one another. So if we're really saying that we should allocate our conservation efforts to these areas shown in red at the expense of those areas shown in green, for example, then we should really be confident as ecologists and conservation practitioners that they are in fact meaningfully different instead of just being lines on a piece of paper. And I can talk about that in the abstract forever, as I often do, but we can also look at some photos of real-life examples of where these ecoregion boundaries are expected to be found. So for example, here I'm showing a photo that I took in northwestern Costa Rica, looking at the transition between a beach, a mangrove forest, and an upland shrubland, all within the scale of about 500 meters. But that contrasts really heavily with the place that I grew up, the northeastern United States, where we can see deciduous forests that seem to go on forever, with the same types of species being found more or less up and down the entire eastern seaboard. So we can imagine places in the world where both of these things are true. And so for that first question, I really do want to ask, at a global scale, do species organize into these geographically identifiable communities, which I'll often call ecoregions throughout the rest of the talk? The second question that I want to ask is actually prompted by that last set of photos that I showed, contrasting the temperate and the tropical zone. A lot of these same thinkers that were summarized in Alexander von Humboldt and Robert MacArthur's work theorize that there might be differences in terms of the strength of these geographic communities depending where on the world you are. So for example, here we have an assemblage of terrestrial temperate species that Alexander von Humboldt, for example, hypothesized that the associations of the same plant species are less consistently extensive and less numerous than in temperate climates, suggesting that in tropics, these communities might be less strong than they are in the temperate zone. While Whitaker, for example, showed convincingly that for the mountains of the United States, the plants appear and disappear independently as we go up a mountain. So Whitaker here is a temperate ecologist and really strongly disputed the idea that these temperate species were clustering, similar to that photo I showed of the eastern deciduous forests. And we can contrast what we see in these temperate zones with what we see in these tropical regions, where Dan Jansen, for example, suggested that mountain passes are higher in the tropics, that these geographic communities are more strongly divided by physical features on the landscapes, such as mountains. And MacArthur suggests that Holdridge, a tropical ecologist principally, might suggest that unlike Whitaker, species do in fact cluster more strongly in the tropics. So we have these competing ideas trading off between some of the largest personalities in the field of modern biogeography. And that's the second question that I want to address, is whether these communities are more distinct in the temperate or tropical zone, whether ecoregions more strongly delimit biological communities in one of these regions. And while there's obviously a lot of conservation implications of these ecoregions today, it's also important to keep in mind that as human beings, we've continued to change the landscape. So this photo looks even more strikingly strange today than it did a few weeks ago. But as I already alluded to earlier, humans have dramatically reshaped the Earth's surface, affecting a wide variety of global change pressures that have all increased the susceptibility of species to go extinct or to dramatically alter their distributions. And today I want to focus on, in my opinion, the two largest drivers of biodiversity loss currently, which are land use change or land use intensification, where we continue to convert natural habitats into either agricultural environments or the built environment, as well as climate change. So in the third chapter, I'm hoping to venture into an exploration of how species and communities will be responding to land use change and climate change, and how that's affected by the ways in which we model those processes. So with that, I'm just going to start by jumping into this first question and thanking a whole lot of people that made this research possible. They were an amazing team to work with and obviously enhanced what I'm about to show today. So this would not have been nearly as polished as it is today if not for them. What I want to do is start by showing this map of ecoregions, which is what I've been alluding to throughout this entire talk. This is a map that was most famously produced in 2001 by Olson et al. And is now widely used by the World Wildlife Federation and the Nature Conservancy, as well as organizations such as the United States Environmental Protection Agency and the European Union to set conservation priorities. But what's interesting about this map is that it relied on almost no data about where species were. Rather, it used maps of temperature and precipitation and rivers and mountains and the opinions of local expert natural historians to inform the drawing of these boundaries. And obviously, local natural history has played a huge role in the formation of a lot of our understanding of macroecological phenomenon. But today we're in an era that Robert MacArthur couldn't have predicted in 1972, where the wealth of biodiversity data that we have is just so immense. We can describe the distribution of plants and animals and their traits and their genes and their interactions across global scales in a way that would never have been possible in 1972. So with this advent of all of these great data sources, can we go back to that question and ask, do we in fact see these ecoregions clustering like species into communities? And the data source that I'm going to focus on today is the Global Biodiversity Information Facility, which is this curated data set where people have gone out to a specific location, here shown in these yellow and increasingly in intensity orange points of where a particular species has been observed in space. And I'm going to overlay that with the existing map of ecoregions that I mentioned from the World Wildlife Fund and from the Nature Conservancy. And what I want to do is essentially recreate what Holdridge and Whitaker and MacArthur and Humboldt would have done, where they went out to a specific location and recorded the plant species and animal species that they found there as they went up a mountain or away from a body of water, and tracked the species accumulation curves and how many new species they were documenting. But since we're going to be doing this digitally, we can do it all over the world. We can do this thousands and thousands of times in thousands and thousands of places. But to illustrate my point, I'm going to zoom into the best place in the world, New Jersey. And I'm going to show here 3 sample ecoregions that we might find in New Jersey. So for example, we have this green, orange, and yellow ecoregion. And what we can do is we essentially drop a virtual naturalist onto the landscape. So we see here in this red dot, and I hope you can see my mouse, but otherwise this large red dot, and the naturalist wanders this path where they crisscross around and sample in this green ecoregion before heading into the orange and finally this yellow ecoregion. And what we can do is record the number of species that the naturalist has seen as they cross this landscape and pose 2 different hypotheses. So the first hypothesis we might pose is the sharp transition hypothesis. And under this hypothesis, we essentially say that ecoregion boundaries form these sharp transition zones where one set of species or one community gives way to another set of species relatively quickly. And if we look at the species accumulation curve that we have under a sharp transition hypothesis paradigmed world, we see that the amount of species that you've discovered increases as you sample within an ecoregion, beginning to saturate as you begin to exhaust the amount of area that you can sample within that ecoregion. But then as soon as you move into the second ecoregion, you all of a sudden see an uptick in the number of species that you're seeing. And similarly, when you cross into this third yellow region, you see yet another uptick in how many cumulative species you've observed. And so another way to think about that is that as you enter each new ecoregion, you see a sudden uptick in the number of new species that you're observing. We can contrast that with the gradual transition hypothesis. And under the gradual transition hypothesis, ecoregions aren't strongly structuring these communities, or rather they aren't describing the structure of these communities. And instead we have species-area relationships that look roughly like these saturating curves that we're very used to in the ecological literature. And there's no strong signal of a number of new species discovery at any of these ecoregion borders. And we can test these theories using statistics and permutation tests, where we essentially take that curve from the sharp transition hypothesis that I showed in the previous slide and fit a stepwise function that's created using the real and known ecoregion borders, where we say this curve intersects with the species area relationship at the breakpoints between the green, orange, and yellow ecoregions, and then we can calculate for this fit line, or this fit stepwise function rather, the amount of the variance in the species area relationship that it's explaining, or the residual. So if it has a really low residual, essentially what that means is the real borders are doing a really good job of explaining the shape of the species area relationship. And we can contrast that to a set of random borders, where we draw these borders on the map relatively randomly, and once again create these stepwise functions, but instead of them fitting nicely to the known ecoregion borders, or these real borders, which are derived from the map that I've been showing throughout the presentation, they intersect this species area relationship at relatively random points along the curve. And we do this not only one time, but 5,000 times, and we can create a distribution of potential residual space. So instead of just having one random border here shown in this dotted line, we do this 5,000 times, and we can generate this histogram which shows the residual from all of these randomly permuted ecoregion boundaries. And then once we have the residuals from those, we can compare that to the residual from the line drawn with the real ecoregion borders. And when we draw this line with the real ecoregion borders, what we can expect to see is that if the residual falls far to the left-hand side of this histogram, below say 95% of those transects, then the known ecoregion borders did a really good job of describing the shape of the species area relationship. We can do that exact same thing for the gradual transition hypothesis example that I posed earlier, once again fitting real and random borders and generating these histograms. But in this case, the real borders, or the borders from the map that was shown earlier, don't do a much better job than any of these random borders in describing the shape of the distribution. So what does that look like when we look at actual data? So let's start by looking at a transect, and again, we did thousands and thousands of these transects across the globe that adheres to the sharp transition hypothesis. So we again see in red, drawn the species area relationship, fit with either the known ecoregion borders in the solid black line, or one example set of randomly permuted ecoregion borders shown in the dotted line. And we can calculate this histogram, which shows here in pink the distribution of these 5,000 randomly permuted bounds, as well as the position of both this dotted set of ecoregion borders and this solid line in terms of the residual amount of variance that they explain. And you can see here the real ecoregion borders have a much lower residual, therefore explaining a lot better the shape of the species area relationship than any of these randomly drawn borders. And we can contrast that with the gradual transition hypothesis, where we fit, again, these real and random borders, and we can see that the histogram of the real and randomly permuted ecoregion borders fall much in line with what we would expect under the gradual transition hypothesis. So as we did this around the globe, there were certainly places that adhered to both of these different elements. So here, for example, perhaps this sharp transition hypothesis came from somewhere like that photo that I showed earlier of the beaches in Costa Rica, while the gradual transition hypothesis might demonstrate a place such as the eastern deciduous forest, where species turn over more gradually. So it's really helpful to look at this as we aggregate across these thousands of transects that we did over the globe. And so I'll do just that for all of these different taxonomic groups that I'm about to show, starting with amphibians. And we see here a strong leftward skew of this density plot. So here you can see that as you move more to the left-hand side of this plot, your data or your transects are more strongly supportive of the sharp transition hypothesis. So on average, for amphibians, the known ecoregion borders had a smaller residual than the randomly permuted borders about 90% of the time, or 90% of the transects. So you can see that this distribution is really strongly skewed to the left in terms of supporting the sharp transition hypothesis and the idea that ecoregions really do strongly cluster amphibians. But it's not just for amphibians, it's for all of these other taxonomic groups as well. And while there's a large amount of variability in how well these ecoregions are describing above ground and below ground communities, there's certainly consistent evidence across these taxonomic groups. I'd like to point out, for example, that fungi appear to be less so well described by these above ground or these ecoregion borders than some of the other animals, yet they still do seem to follow some of these same global biogeographic patterns. And that's obviously something that's really interesting and I dive into in more depth in other places, But today I just sort of want to answer this question relatively simply and ask whether species do organize into geographically identifiable communities. And I think here we show compelling evidence that in fact they do. And just to show two examples of what that looks like in the real world, the photo that I've already shown of the beaches in Costa Rica, set next to a photo in the Andes where we see the sharp tree lines as you move up in elevation where where woody plants are quickly replaced by grassland species as the elevational tree lines set in. So just to move back to this questions overview slide, after showing evidence that species do in fact organize into these geographically identifiable communities, I think it's logical to then transition to the second question about whether or not these communities are stronger in the temperate zone or in the tropical zone. And once again, I owe a huge amount of thanks to these great co-authors and collaborators on this project. And for this, I again want to take us back to this map of ecoregions, which I've been showing throughout the talk, but I want to do somewhat of a different analysis to answer the question than we did in the first talk. So to illustrate this analysis, let's take 3 of these ecoregions and just turn them into blobs. So we now have these 3 relatively generic looking ecoregions, but we know a lot about each of these ecoregions. For example, we know their temperature, with this ecoregion being warmer than this ecoregion, or the precipitation, with this ecoregion being wetter than this ecoregion, the amount of tree cover they have, the amount of human land use they have, and all of these other different attributes that are associated with potentially structuring communities across space. But we also know a lot about what species are contained within those ecoregions. So here I've illustrated a suite of species with green squares and yellow triangles and blue stars, some orange diamonds and gray hexagons thrown in for good measure. And we can compare the communities that are found within these different ecoregions. So if we start out by comparing these 2 ecoregions, we can see that they share a lot of the same species. In fact, only the orange diamonds are different between these 2 ecoregions. So they have a relatively low level of heterogeneity when we compare them. So I'm going to call that this low external heterogeneity. But when we compare, say, this ecoregion over here with this ecoregion on the right-hand side, we can see that while they share some species, there are more species that are new between them. So for example, the blue stars are present only here and not in this ecoregion. But if we then go to compare these final 2 ecoregions, they in fact have a high degree of external heterogeneity, with only the green squares and the yellow triangles being shared, while the blue stars, orange diamonds, and gray hexagons are unique to one of those 2 ecoregions. And what we can do with this information is essentially ask how much of this external heterogeneity can be explained by the environmental conditions within each of those ecoregions, the amount of human land use that they have in that ecoregion, and then, of course, we will be controlling for the taxonomic group of interest, as well as the geographic distance between those ecoregions, just to make sure that we're not obscuring any potential patterns because of these obvious covariates that we should be accounting for. What I'm quickly going to do is to give you an overview of the graphs that I'll be going through in this section, just put up this blank framework and walk through it step by step. So here we have a relatively important variable of deciding whether or not you're in the temperate zone or the tropical zone, mean annual temperature. So we can see here on the x-axis, we just have a normalized value, and we can outlay places that are colder and warmer, here representing temperate or boreal zones, and here tropical zones. And then on the y-axis, we can ask, what does that do in terms of how dissimilar communities or ecoregions are from one another? So more positive values indicate that ecoregions are more strongly grouping communities, while negative values indicate that the communities contained in those regions are less distinct from one another. And once again, we can do this across our 7 different taxonomic groups, and we can plot out the relationship between mean annual temperature and how distinct ecoregions are. And you can see across almost all of these taxonomic groups, again with the exception of fungi, which I alluded to in chapter 1, there seems to be this really strong upward pattern where warmer areas contain more distinct communities and colder or cooler areas contain less distinct communities. And so I think we can show here that ecoregions are more dissimilar from one another in warmer regions. There's also a lot of other ways that we can think about what differentiates the tropics from the temperate zone. One of them, for example, being temperature variability. So in temperate zones, we have relatively cold winters and warm summers. So we have this strong seasonality of temperature. The temperatures over the course of the year are highly variable, while in tropical areas, there's very weak seasonalities. So there's not a strong swing in the temperature between the various parts of the year. And we can once again compare this to the distinctness of these communities in these different areas. Plotting out here for these different taxonomic groups, we once again see that there's strong seasonality leading to more similar communities, while weak seasonality leads to more distinct communities. So this once again suggests that in these ecoregions that have attributes associated with being tropical, such as high temperatures and low variability in temperature across the course of the year, we see more distinct communities. But obviously that's only one component of this. And the question that I put at the top is whether or not communities are more distinct in the temperate and tropical zone. So while I can put a palm tree and a pine tree up, I think the only way to really conclude about whether or not ecoregions are more distinct in the temperate or the tropical zone is to actually look at this on a map. So when we plot out the expected dissimilarity between ecoregions, we see here that more or less these more dissimilar ecoregions shown in yellow are almost exclusively clustered around these tropical zones, while these less dissimilar ecoregions are in the temperate and boreal zones, showing fairly compelling evidence that ecoregions are more dissimilar from one another in the tropics. And that sort of simply answers the second question about whether or not ecoregions are more distinct in the temperate or tropical zone, with the tropics usually containing more distinct communities from one another. And now what I want to turn to is this final question of how patterns and dynamics of biodiversity are likely to change with climate change and land use intensification. Once again, starting by thanking an amazing group of co-authors that have been super valuable to this project. And for this, instead of focusing on all of these different taxonomic groups, I'm going to focus in on one taxonomic group that I've had somewhat of a complicated relationship with, and that's birds. So for those of you who know me and what I tend to study, if I principally am focused on insects, and birds eat lots of insects, then they obviously aren't necessarily my best of friends. Moreover, I find that too often people who study birds get all the attention. So for example, when Nick Hendershot and I, who is defending his dissertation tomorrow, if you're interested more in birds, you can come hear him talk all about them, would discuss what we were studying with people at dinner, I would often go first and say that, oh, I study insects and how they change as a function of how much natural habitat is in these different coffee farms, blah, blah, blah. And then Nick would just say, oh, I study birds. And then they'd proceed to just turn and talk to Nick for the rest of dinner, perhaps leading to my inferiority complex, where I'm not as fond of birds as I am of some of these other taxonomic groups. But rest assured, there are many birds that are quite photogenic and quite elegant, and they also are an amazing organism to study, or an amazing set of organisms to study, just because of the immense amount of data that we have on how they're distributed across landscapes. So here I'm showing the resplendent quetzal, which is one of the most iconic birds, not only in Costa Rica, but the entirety of the world. And the amount of enthusiasm surrounding birds has led us to be able to generate an immense amount of information about where they're distributed across landscapes. So here I'm showing data from the Global Biodiversity Information Facility, or GBIF, about where the resplendent quetzal's been observed across Costa Rica and then into Panama. And we can compare this to various patterns of temperature and precipitation and try to get a better understanding of how these climate and other patterns are driving the distribution of the quetzal across the landscape. And so as you remember in chapter 2, temperature and precipitation and all these other attributes that are associated with the tropical zone or the temperate zone can really strongly dictate where we find different sets of species or different communities. And in fact, that's the common approach that's used in what's called the species distribution modeling literature, where we pair information from the Global Biodiversity Information Facility or some other data set with climate conditions such as temperature and precipitation, and employ these statistical models called SDMs, or species distribution models, to create maps of nationwide prevalence. So here we can see in these light, higher intensity colors where the quetzal is more likely to be found, and in these black or darker colors, areas where the quetzal is less likely to be found. And this has greatly informed our understanding of macroecological processes. But the problem is that these models typically don't tell us that land use is really strongly structuring species communities. So for example, if we think back to chapter 2, I was showing how the dominant driver at a global scale was the distribution, or rather the patterning of temperature and precipitation from the tropical to the temperate zone. But when you go out in a landscape in Costa Rica, for example, this photo that I took in Monteverde, we can see that all of a sudden, while temperature and precipitation over a small area might be relatively homogeneous, the landscape characteristics are incredibly different. So if we look at this photo, we see a large patch of remnant forest in the background, but in the foreground, we see elements of the built infrastructure with a road winding its way through this photo, with houses and cattle pastures interspersed with hedgerows and these other remnant forest elements. And what we found as a lab group when we've gone out to these landscapes is that that has a really strong impact in the number of birds that you find in a given habitat. So if we look at comparing bird species richness to how much forest cover is surrounding one of these fields, we all of a sudden see that changing the amount of forest cover between 0 and 100% can change the number of species that you find there by a factor of 2. So you might go from having about 80 species to almost 180 species. And this speaks to this other component of how biogeographers describe species across landscapes, focusing on local habitats and incorporating high quality data where someone has gone out and counted the number of species that they observe in a place, noting what species aren't there, as well as really locally defined characteristics of the habitat or the vegetation that are likely to be important for how species are changing across an area. And they calculate how that influences the abundance and richness of birds. So what I want to do is essentially take those two different approaches. The first approach that I showed in chapter 2 at a macroecological scale that well describes the distribution of species in response to climate, and combine it with what we know from these local studies, which suggest that local habitat availability is hugely important, into a single model. So on the left-hand side of the model, I'm showing here this workflow that I described for these global species distribution models, and pairing it on the right-hand side with the general models that are used in these local studies, and combining them into the single model, which is able to capitalize on the macroecological strength of species distribution models with the local strength of describing communities across well-documented patterns of vegetation and impervious land surface and bare ground and all of these other habitat elements that species are actually utilizing. And so what I want to do is essentially compare these approaches across, as Gretchen said, Costa Rica, which has been a huge focal study system for my dissertation, and something that I'm disappointed to not be able to go into more detail today, but I've spent a lot of time in Costa Rica, both in Guanacaste and in Colabus and in these other regions, looking at the distribution of birds and insects across conditions. And here I'm utilizing a set of data collected by Jim Zook over the past 20 years, where we've set up these bird sampling transects, 48 of them, in these 4 distinct regions across Costa Rica. So you can see here these clusters of red transects, where Jim has gone out and counted the number and identity of all of these different bird species for the past 20 years. What's great about this setup is that since we have these 4 distinct regions, we have this variability in climate in terms of the temperature and the precipitation and the seasonality to describe how climate is changing the occurrence of these species between the regions. But then within each of these regions, for example, shown down here, we see that there's some of these transects that are situated in much stronger or much wider extenses of forest cover, while some are situated in these relatively open areas. So when we compare across the transects within a region, we can compare those local habitat availability elements to understand at a large scale how climate is influencing these patterns, but then at a local scale, how land use is structuring where the birds are actually found. And we can plot out the relative importance of climate and land use to describing the distribution of species across space. So here I'll show the results from the standard species distribution models that include both climate and land use, and showing here on the x-axis how sensitive they are to climate, and on the y-axis how sensitive they are to land use. So if they're more sensitive to climate, the values on the x-axis should be higher than the values on the y-axis. And I'll also note, by definition, these have to sum up to 100. When we plot this out in space, we see that the 271 species that we modeled, 268 of them have a higher sensitivity to climate, while only 1 species is more sensitive to land use, shown in red. I'll note that there's 2 points shown in gray that we can't differentiate statistically as to which is a more important driver for their distribution across the landscape. And we can see here, for example, the bay-headed tanager, which I'll come back to later in this slide, has almost 95% of its variability explained by climate, or rather 95% of the explained variance is driven by climate, suggesting that global standard species distribution models imply that climate is the overwhelmingly dominant driver of species distributions. And that's certainly what I showed in chapter 2, with the difference between the temperate and the tropical zone really strongly driving patterns in community structure across space, suggesting that this is in fact in line with the standard thinking of how macroecological patterns play out. But when we contrast that with a hybrid model that's constructed using both these global approaches, but also these local data products and these high quality bird abundance data sets, we see a really different picture. All of a sudden, only 6 of our 271 species are more strongly driven by climate, while 201 of them are more strongly driven by local land use. There's a lot of species that we can't differentiate which one is more so driving their presence or absence across the landscape. But essentially, I think here, the main take home is that fine scale land use and biodiversity data show an outsized importance of human land use. And obviously we can think about this in the context of what's happening in these 48 sites that Jim Zook's been going out to visit, But it's also really interesting to see what that means when we project out into either time or space in terms of how species richness is structured across landscapes. So I'm going to first show a map of the country of Costa Rica and the expected bird richness using standard species distribution models. So some things pop out immediately here. We have higher richness along the coastline, lower richness in this central mountain range, and lower richness in the Nicoya Peninsula and in Guanacaste than in the rest of the country. And when we compare that to the predicted richness from our hybrid model, which incorporates this local land use, you can see that to a first approximation, the maps look basically the same. We once again see higher richness along the coastline with decreased richness in the mountains and in this northwest region. So these standard models are comparable to our hybrid model in terms of describing these present-day macroecological patterns. But where this becomes more problematic is when we contrast the expected responses of these species and communities to various climate change and land use change scenarios. So for example, if we look at the impact of climate change on species in our standard model, we see that there's certainly spatial variability in which climate change is expected to have a higher or lower impact, but this impact is still expected to be relatively large with a per pixel change of about 8.1%. And I'm obviously happy to go into more detail later about what that figure represents, but for the sake of time, what I want to do is just track on the side of all of these maps the relative impact of these changes based on this percent change from the baseline. So just compare these numbers as we go throughout the scenarios to see what the relative impacts of climate and land use change are in these different scenarios and these different models. And so we can contrast that with a deforestation scenario in which about 5% of the tree cover is lost in these individual habitats. And we can see that there's certainly, again, a strong spatial patterning and a notable effect of deforestation on the species and communities that we find in these different habitats. But it's only about a third of what's expected under climate change. And that's relatively in line with a lot of the global modeling efforts, which suggest that by 2050 and by 2070, climate change is going to have a much larger impact on global biodiversity than will land use intensification or land use change. However, if we compare that to our hybrid model in terms of its impacts, projected impacts of climate change and land use change, we once again see that the climate change scenarios are relatively impactful for biodiversity, with the magnitude of the effect being about on par with what's expected under the standard model and showing similar spatial patterning to the standard models. But where we see a really pronounced deviation is if we compare to land use change scenarios, all of a sudden we maintain the spatial patterning, but the impact of local land use alterations on biodiversity have doubled in intensity. And so once again, this is a relatively mild deforestation scenario, which indicates that perhaps under more robust deforestation pressures in other regions of the world, our standard models and our standard ways of describing species distribution models may be underestimating the impacts of land use intensification. And that's sort of the main take home that I'd like to share here. But I'd also like to spin it in another direction. And instead of just saying that there are inevitably going to be these negative impacts of land use intensification, also highlight the positive message that this sends, saying that relatively small changes to the habitat availability to different species on local scales can have impacts that are on par with the impacts of global climate change. So all of a sudden, these things that are being driven, we think, by these global actors that we couldn't possibly have any impact on, can actually be offset by these relatively small scale restoration actors that we as individuals or as small management groups can take. And to conclude, I'd just like to summarize what I found over the past 3 chapters. First, asking whether communities cluster into ecoregions, and I think showing that ecoregions do in fact effectively delineate biological communities. When we turn our attention to whether or not they're more strongly delineating communities in the tropical or in the temperate zone, it appears that ecoregions better describe tropical communities. But finally, when we turn our attention to land use and climate change, it's really a reliance on our models and data that suggest that climate change at a global scale is driving all these patterns, while we know that incorporation of local land use data can really show an outsized importance of habitat availability. And I want to return finally to another quote from Robert MacArthur's 1972 book, where he said that no one had carried out in the tropics a study like those of Whitaker. And again, Whitaker was the temperate ecologist who walked up these mountains. And we must await such a study before we can pass final judgment on whether life zones are real in nature or whether they are the scientist's convenient but arbitrary classification. And I'd say here that I've effectively showed that life zones are in fact both real and incredibly convenient. And convenient here being a management term that we're using to say that we can really effectively describe where communities are and how to best conserve them. And to conclude, finally, I'm going to turn briefly to some of the future directions that I'd like to pursue, both scientifically and in terms of the application of this work to other things. So as I move forward in my career, I'm really motivated by two main questions. First, why are things where they are? And I think today I've given you an overview of how that shapes the distribution of ecoregions. I've worked a lot on the density and distribution of trees across space. I've also started to work more on birds. Oops, wrong bird photo. Here's one of me pretending to like birds. Once again, if you're around tomorrow, please come check out Nick Hendershot's talk, and you'll hear a lot more about birds than I was able to go over today. And of course, I'm really distraught to not have been able to cover more about the distribution of insects across landscapes. The second question that I would hope to answer, or continue to answer rather, is why does it matter where those things are? So that sort of summarizes a lot of geography, biogeography, and questions that are easy to comprehend. And here I'm thinking a lot about the functioning of ecosystems. So for example, the role of insects and other small mammals in grassland food webs, the relationship between biodiversity and carbon sequestration, or the provisioning of fresh and clean water across space and time. But then also a lot of ecosystem services that we as human beings depend on for our well-being. So a lot of our value that we're deriving from nature comes from our interactions with biodiversity and other types of nature or ecosystems. So for example, ecosystems provide large opportunities for ecotourism. So for example, working on this paper with a great undergraduate student who was exploring patterns between bird biodiversity and wildlife-based tourism in Costa Rica. Thinking about the mental health benefits of constant and sustained interactions with nature. And sort of in general, thinking about the role of biodiversity in providing ecosystem services that enhance our well-being as people. And so making the case for conservation, not only from an intrinsic perspective of why biodiversity should be important, but also a really practical perspective of why biodiversity enhances our sense of value as people. And so to just talk briefly about that last point, thinking about the policy relevance of this work, I'll highlight two projects. The first work in Costa Rica that we're undertaking with the Central Bank and the Ministry of Environment and Energy, linking these essential biodiversity variables. So here, ecosystem level biodiversity variables such as tree cover or the distribution of ecosystem types across the landscape, along with species level essential biodiversity variables such as the distribution of birds or the distribution of pollinators, to generate maps of ecosystem services across the entirety of Costa Rica and providing those to the central bank so they can help aid decision makers in understanding the financial benefits of having these natural elements across their landscape. While there's obviously some examples of where the benefits of biodiversity and nature align with financial benefits, there's also sometimes when there's trade-offs that are necessary between the development of our landscape for other purposes and the protection of biodiversity or other types of nature. So in that case, what we want to do is be smart about the trade-offs between any type of production that we'd like to undertake and the amount of species that are likely to be able to persist in those altered landscapes. So providing guidance here in a project with the World Bank to better understand how we can manage these trade-offs effectively. And so just to close, 2020 is set to be the next major meeting of the Convention on Biological Diversity in October or November of this year. And a lot of this work, I hope, can help us refine a vision for how we hope to sustain our lives into the future, living harmoniously with nature. And with that, there's a ton of organizations that I'd like to thank that provided help or funding for this project, and I think we can turn quickly to questions. Thank you.
Okay.
Thank you all for tuning in today. And in particular, I want to highlight some people that I need to thank. First, I owe a huge amount of thanks to my advisor, Gretchen, who obviously you've all now met, who's been such an amazing traveling companion in the field, in the lab, in professional settings, and in these more recreational and social settings. And I just I don't see any way in which any of this would have been possible without your guidance throughout all of these various stages, from our first field season in Costa Rica up in the top left, all the way through to today's presentation. And I'm just eternally grateful to you. But it's obviously not just one person who shaped my academic career. I owe a huge amount of thanks to my committee today, Dr. Fukami, Pei, Mordecai, and Gabrielle Wong-Parodi, for helping out and guiding this process, as well as a lot of faculty from other institutions. Aaron, you might recognize yourself trying to throw a washer up in the top left photo. I just felt the need to highlight that. And then also, as I went throughout the entirety of this presentation, I showed photos of all of my collaborators that helped with these various chapters. I'd also just like to give them a shout out here. Not only were they amazingly helpful in shaping a lot of the science, but they were also incredibly fun people to be around outside of the lab, and I owe them an immense amount of gratitude. I'd also like to thank the Stanford Center for Conservation Biology and Natural Capital Project for being such a great home to do this work in. Working with you people every day has been such a great pride, a great sense of joy coming to the office, and just seeing you all around was amazing. I also owe so much thanks to all of my collaborators in Costa Rica for making the field station feel like a home away from Stanford and being such an amazing place to be carrying out a whole chunk of my dissertation research. And also on a more personal note, I want to turn back in time and thank a lot of my teachers throughout elementary school and my undergraduate and master's experience that have really guided a lot of who I am today as a person in terms of inspiring a love for science and the environment. And I also owe so much thanks to so many friends who have really shaped who I am as a person, and I know I'm eternally grateful for. And then lastly, just to thank my family. They've been there from the very beginning, as you can see in the top left photo, and have been incredibly supportive throughout this entire process. And especially my parents and my sister, who I hope are still here, and thanking them for all of what they've done and just constantly supporting me, not only over the course of my PhD, but for as long as I can remember. And I'm just so, so thankful to you all. And then lastly, to my dog, from puppy to who he is today. And I want to thank you all for listening in and just really appreciate you taking some time out of your day.